Welcome everyone to our SCIEX Biopharma vodcast. I'm Kerstin.
And I'm Roxy. And in this series, we're uncovering how scientists are rewriting the rules of analytical science in biopharma.
In today's episode, we have Thomas Kofoed with us, the CEO from Alphalyse. Welcome, Thomas.
Thank you.
We are very grateful to have you here, Thomas. Where in the world are you right now?
I'm actually situated in Denmark.
That's very cool.
All right, this is our quickfire round. So we'd like to get to know a little bit more about you. We're going to ask you a series of questions. If you could answer with the first thing that comes into your mind. Ready?
Yes, I'm ready.
As a scientist, Thomas, are you rather a gatherer or hunter?
I'm a hunter.
Thomas, do you prefer easy analysis or a difficult one?
Oh, I'm always going for the difficult ones, but I want to make them easy.
Good answer. All right, we will jump into some more longer questions. Thomas, can you give us an overview what HCP analysis means? What is that?
Yes. As you know, most of the biologics that are produced and that are very successful as a drug, they are produced in typically in host cell organisms. And when you, of course, purify your drug molecule or your drug molecules, you remove a lot of the host cell proteins as part of the purification process. But no matter how well you do that, there will always be proteins left from the host cell. And you need, of course, to document and show that the level of these host cell proteins are at a certain level. And actually, we think it's also a good idea to know exactly what host cell proteins do you have in your drug substance.
And why is it important to perform analysis of host cell proteins?
Yeah, host cell proteins can have properties that can be really bad, either for the stability of your drug product. They can also be immunogenic, so they might cause problems once you start providing the drug for patients. So you would like to know exactly what host cell proteins do you have and predict and see if those might be of concern.
Yeah. Now, how does HCP analysis of antibodies differ from that of viral vectors?
Yeah, typically antibodies are purified by using protein A purification, and that makes them, in general, very clean products. Whereas cell and gene therapies, they are typically much more complex to analyze and also contain a lot, not only host cell proteins, but also proteins from the viral vector itself, the viral protein, and other sources. So typically they are quite complex compared to monoclonal antibodies.
Yeah. And then if you had one wish to shape future technology for HCP analysis, what would that be?
I think I would like to speed it up, so it's still a quite long process. So we spent hours analyzing each samples, also because we don't do them in triplicates. And then it's of course related to software. So I think the software can still get better. And we would also like to have software that can be used in a GMP environment, because that's where we are moving towards GMP analysis.
Thomas, in general, what was your motivation to focus on HCP analysis?
Yeah, we have used mass spectrometry to characterize biologics for many years. And we actually saw that some of our clients were using our service to look at the host cell proteins. So they ran 1D and 2D DLs and cut out bands and asked us to analyze those bands and identify the proteins. And then we are very curious, so we thought, what is it actually they are doing? And we could then see that the limitation of the current technology, the ELISA technology, was that mass spectrometry could provide much more detailed information. So that's why we started with the focus on our clients, how we could see that mass spectrometry could help them. And then once you start using mass spectrometry for this, you just get excited and can see how much more information it provides and what kind of details and how much you help your clients
So, Thomas, what advice would you give future scientists so that they can keep pushing the boundaries with new technologies?
I think I often heard this, people saying this is not possible. A lot of scientists, they have a tendency, they know a lot and they can then plan a lot, and then they typically also can shoot it down again and say, yeah, this might not work. And I would say with the analysis we have established for host cell proteins, I've been really surprised about how reproducible it's been. And if I had started saying this to scientists, they would have said, no way, it's never going to be possible to do what you are doing. So I would not listening too much to people, and then just try it out and see if you can solve it as you go.
Yeah, that's excellent advice.
Thomas, our vodcast is titled Rewrite the Rules. What do you think about how that applies to what you are doing?
It fits perfectly well. I don't know if you've seen it, but just this Monday, the new chapter came out in the US 𝗥𝗲𝗄𝗶𝘁𝗲 about using mass spectrometry for host cell protein characterization. And I think for many years, ELISA has been the only way of doing this. And now with this new chapter, I think it's going to grow even more. And it is kind of rewriting the rules that by using mass spectrometry, you now get much more detailed information about these important impurities that you need to understand to bring new safe drugs on the market.
Awesome answer. Thank you. Thank you so much, Thomas, for joining us today and sharing your insights.
Okay, thank you. Pleasure to join.
Bye, Thomas. And to our viewers, thank you for watching. Be sure to visit SCIEX.com to learn more about our applications for HCP analysis. Until then, keep rewriting the rules. Goodbye.
Goodbye.